Hello, welcome back friends. In this video we will going to discuss about hemagglutinin in a very detailed manner. So what is hemagglutinin? Hemagglutinin is a glycoprotein which is present on the surface of the viral envelope of influenza, right? So this hemagglutinin play a important role in the entry of the virus into the host cell. Okay, without hemagglutinin the virus cannot enter. Okay, and that is why most of the neutralizing antibodies try to target hemagglutinin. Okay, so influenza virus, first of all, how mediates their entry? Let's see. Okay, so we all know that influenza virus consists of two type of glycoprotein. One is hemagglutinin, another one is neuraminidase. Clear? So in the surface of their target cell there is a special type of receptor present which contains polysaccharide terminating with sialic acid. Okay, that is known as sialic acid receptors. So hemagglutinin will interact with those sialic acid receptors. Okay, that is how they mediates their entry. Okay, now in after entry they will be there is a receptor mediated endocytosis by which they enters inside the host cell, and as a result inside the host cell they will be surrounded by vesicles. Okay, now after that, wait a minute. Okay, so now after that there will be a fusion called endosome lysosome fusion. Okay, that is the endocytic vesicle which contains the virus fuse with lysosome. Okay, and lysosome contains digestive enzyme and acidic interior. Okay, so under such circumstances this digestive enzyme have the ability to digest the things that the host cell taken in. Okay, now in case of influenza virus there is a bad news for the host cell, that the acidic interior that the lysosome provides, the influenza have the mechanism to use those acidic interior for their uncoating process. How? Let's see. So now this is the structure of the influenza virus. So inside the host cell they will be covered with a so they will be covered with a endocytic vesicles. Okay, now lysosome will come which contains, let's say, digestive enzymes and let's say some acidic interiors. Okay, so now when they will fuse those acidic interior, that means the protons will be taken in by the M2 ion channel. Okay, and these protons will allow the disruption of the protein interaction, causing the matrix protein to detach from the viral protein that covering the viral RNA genome. Okay, and that is the beginning of the uncoating process. Clear? So after entry inside the host cell they will be surrounded by endocytic vesicles, right? So inside that there will be viral envelope, right? And inside the viral envelope there will be matrix protein, and inside the matrix protein there will be another protein, and then your genome, which is 8 segmented RNA genome, right? So there will be another thing, that is lysosome. This lysosome consists of digestive enzyme and acidic interior. Okay, so when lysosome fuses with endocytic vesicles, the acidic interiors, that means the protons, are taken in by M2 ion channel. Okay, so then these protons will help help to disrupt the protein interaction. Okay, and as a result, the matrix protein will be detached from the viral protein that covers the RNA genome. Okay, and this detachment is called the beginning of the uncoating process. Okay, now let's try to think about this. If we somehow block the ion channel, that is M2 ion channel tetramer, can viral genome can release into the cytoplasm? No, there is no possibilities, right? So that is why there is a antiviral drug available which specifically targets the M2 ion channel, named amantadine and rimantadine. Okay, rimantadine also known as adamantins. Okay, so now after that we have begin the uncoating process, but this is not enough. Okay, now what virus will do, not virus, hemagglutinin. Before going to talk about this, it is important to talk about the structure of the hemagglutinin. Before that, I want to told you that I think you remember that at the beginning of the video I have told you that hemagglutinin targets receptor containing polysaccharide terminating with sialic acid, right? So there is a special type of recognition site present in between sialic acid and a galactose, which is the polysaccharide present in the receptor. So that particular recognition site is recognized by the hemagglutinin. Okay, there can be alpha 2,3 linkage between sialic acid or galactose, and alpha 2,6 linkage between the sialic acid and galactose. Okay, so and different type of hemagglutinin present in different influenza strain have the ability to recognize different linkage. Okay, for example, alpha 2,3 linked sialic acid and galactose linkage will specifically recognized by hemagglutinin, which is present in H5N1 strain of the influenza virus. That means the strain which is present in avian influenza virus, right? So but this type of linkage, that is your alpha 2,3 linkage, is present deep within the respiratory tract of the human. Okay, and that is why H5N1 strain, despite very deadly, they are rarely transmitted from human to human. Okay, on the other hand, in the upper respiratory tract, alpha 2,6 linked receptor are present, and which is especially recognized by influenza A, and that is why we are most of the time infected by influenza A. Clear? So now this hemagglutinin is not a single subunit, it is a complex of trimer. Okay, or more specifically saying homotrimer. Okay, so each of the subunit consists of two things. One is fusion peptide part and another one is receptor binding part. This part will bind with the receptor and this part will the fusion peptide. Okay, and now for a virus to become infective, the first step is cleavage of the hemagglutinin, or cleavage at the particular position of the hemagglutinin. Okay, that will expose the fusion peptide. Okay, and the enzyme here is unfortunately a host cell enzyme which is coming from the epithelial lining of the respiratory tract. Okay, that is what I have written here. For an influenza virus to become infective, hemagglutinin must be cleaved by an enzyme and it is released from the epithelial lining of the human respiratory tract. Okay, and this cleavage frees one end of the segment called fusion peptide. These peptides are hydrophobic and buried within the core of the hemagglutinin trimeric complex. This fusion peptide will going to play a very interesting role later. So that is what we will going to talk about. Okay. So if you remember, I have told you that after the beginning of the uncoating process, due to the low pH, there will be something that will ensure the uncoating. Okay, so the idea is here, due to the low pH, hemagglutinin will undergo a conformational changes. Okay, the receptor binding domain will move back and the fusion peptide will move towards the vesicle membrane. Okay, and many trimeric hemagglutinin molecules at the same region of the membrane, with the help of this fusion peptide, try to fuse with the vesicle membrane. Okay, clear? So what is going on here? First of all, the domain which is involved in receptor binding move back. Okay, and the fusion peptide will move towards the vesicle membrane. Suppose this red lining is the vesicle membrane. Okay, and now what will happen? Let's say if this is a host cell, now let's say if it is your virus, so near many hemagglutinin molecule together try to fuse with the vesicle membrane. Okay, and as a result, the vesicle membrane and okay, so we have already entered inside the virus, so it will be not the virus, instead it will be your endocytic vesicle. So inside the endocytic vesicle there is a virus present, right? Now the viruses that, let's say this is one hemagglutinin, this is another, this is another, so together 3 hemagglutinin trimeric complex together try to fuse with the endocytic vesicle. Okay, and how this is occurring? This is occurring due to the low pH, which causes a conformational changes in the hemagglutinin, and as a result the receptor binding domain move back and the fusion peptide move towards the vesicle membrane. Okay, and a number of hemagglutinin molecule together try to fuse with the vesicle membrane. Okay, and as a result all the viral content that is present will released into the cytoplasm. Okay, so that is the overall function of hemagglutinin. Now if someone told you to write about the function of the hemagglutinin, you have to mention the entry of the virus, how they interact with sialic acid by recognizing the linkage in between sialic acid and galactose. Okay, and then you have to write about this thing, that is how due to the conformational changes of the hemagglutinin, fusion peptide try to fuse with the vesicle membrane and ensures the release of the viral content in the host cytoplasm. Okay, clear? So for a quick recap, hemagglutinin is a glycoprotein receptor, okay, or a glycoprotein molecule which is present in the surface of the viral envelope of the influenza virus along with neuraminidase. Next video will be all about neuraminidase. So this hemagglutinin have the ability to interact with the host cell receptor that contain polysaccharide terminating with sialic acid. Okay, so why they are recognizing this kind of receptors? Because hemagglutinin have the ability to recognize a special type of linkage in between sialic acid and galactose. This linkage is known as alpha linkage. Okay, if alpha 2,3 linkage is recognized by the hemagglutinin, make sure your virus is H5N1, that is avian influenza strain, but avian influenza strain cannot infect humans because those kind of linkage are present deep in the respiratory tract. Okay, that is why it is quite difficult for them to reach there. Instead, the upper respiratory tract contains alpha 2,6 linked receptors. Okay, and that is mostly recognized by our influenza A viruses. That is why humans are mostly susceptible to influenza A viruses. Okay. Now, after that recognition, virus will be enters inside the cell via receptor mediated endocytosis. Okay, and as a result the virus are surrounded by vesicles, that is endocytic vesicles, right? Then there will be fusion of the endocytic vesicles with lysosome. That lysosome contain digestive enzymes and acidic interior. This digestive enzyme have the ability to digest what the host cell has taken in, right? But here the influenza virus uses these things for their own advantage. For example, the acidic interior, that means nothing but the protons. Influenza ingests all these protons via its M2 ion channel tetramer and uses these protons to detach the viral matrix protein from the viral protein surrounding the RNA genome. Okay, that begins the uncoating process. Now, this low pH causes a conformational changes of the hemagglutinin. Okay, and as a result the receptor binding domain goes back and the fusion peptide moves towards the vesicle membrane. Okay, and now with the help of this fusion peptide, near about many hemagglutinin together try to fuse with the vesicle membrane. Okay, and as a result the viral content inside the influenza virus expels into the host cell cytoplasm. Okay, and that is how the entire thing is going on. Okay, so I hope this concept is now very clear to you. So thank you for listening to this class. In the next video we will going to talk about another glycoprotein present on the surface of the viral envelope, neuraminidase. Okay, this neuraminidase will help viruses to release from the host cell. So thank you for listening to this class.